Welcome to all. Today we see about the protein classification. First we see about the definition of protein. The proteins are one of the food component like carbohydrate. The proteins are macromolecule, organic molecule, and biomolecule, which means that is present inside of all the bioorganisms. The proteins are linear chain of amino acid that are linked together by covalent peptide bond. That means the protein contains so many amino acids. The 20 amino acid of our body that is repeatedly joined by the covalent bond that is known as protein. Each protein has specific and unique structural output and a sequence of amino acid that defines both its three-dimensional structure and its biological function. Then we see about the classification of protein. They are classified on the basis of their function and the physical and chemical properties. First, and detailedly, we know about that. We must know about that the classification of protein based on functions. In a functional classification, they are grouped into six. First, the catalytical protein. Catalyt Catalytical means all the enzymes are made by proteins, so all the enzymes are under this category. So in all the proteins are not enzyme, but all the enzymes are protein. That is the role. So all the enzymes of our body that have the catalytical role, which means they take over some of the chemical reactions. They are all called as enzymes. First, glucokinase, dehydrogenases, transaminases, and many more enzymes. So many enzymes are present in our body that under the catalytical protein. Which means the catalytic means the proteins are enzymes that is role under catalyst that enhance or speed up or accelerate any of the reaction of our body, so that is known as catalytical protein. Next we see about the transport protein. Like the transport, the proteins are involved in process of transportation. That means which transport one's of any chemical substance from to another area. Example, hemoglobin transport oxygen and carbon dioxide, commonly the transport gases. Next, transferrin transport iron, albumin carries fatty acid and bilirubin. Next, we see about storage protein. Many proteins serve as storage form. So the proteins have stored many of the chemical substances, especially ions. Example, apoferritin stores iron in the form of ferritin. Myoglobin stores oxygen in muscle. Next, we see about contractile protein. Some proteins have the ability to contract and function in the contractile system of skeletal muscle, like a contracts. So, example, actin, myosin. Next, structural protein. Many proteins serve as supporting framework of cells to give biological structure, strength, or protection, rigidity to the cells and tissues. So, examples collagen in bone, cartilage, elastin of ligaments, keratin of hair, nail, etc. Next, defense protein. Many proteins involved in defense mechanism against invasion of foreign substances such as viruses, bacteria, and a cell, which means some of the foreign substances around the atmosphere so that attack our body in any time, so they are killed by the proteins that are called as a defense protein. Example, immunoglobulins are antibodies. All the antibodies are protein. Fibrinogen and thrombin are blood clotting proteins that prevent the loss of blood when the vascular system is injured. So these are present inside of plasma. The next, regulatory proteins. Some proteins regulate some of the cellular and the physiological activity, which means the reactions. Example, many hormones example, insulin and glucagon that regulate the sugar metabolism, and growth hormones regulate the growth. Growth hormones of pituitary gland regulate the growth of cells. So there are six groups of proteins we studied about under the under the physical sorry under the functional classification. Okay, next we see about the classification of protein based on physical and chemical properties. According to the Joint Committee of American Society of Biological Chemists and American Physiological Society, the proteins are classified into three: simple protein, conjugated protein, derived protein. First, under this simple protein, the simple proteins are defined as those proteins that upon hydrolysis yield only amino acid or their derivatives, which means when hydrolysis the proteins can undergo some amino acid or their derivatives. This is subdivided based on their solubility and the heat coagulability. That means the soluble nature and the coagulation nature, they are classified further. First one, albumin. Albumin is the major or predominantly present in plasma, that is soluble in water, coagulated by heat. The glycine amino acid, it is not present in albumin. Example, egg albumin, serum albumin, lactalbumin of milk. Next, we see about globulin. The globulins are insoluble in water, but they are soluble in dilute neutral salt solution and are heat coagulable. Ovaglobulin of egg yolk, serum globulin, myosin of muscle. Next, we see about glutalin. The glutalins are soluble in dilute acids and alkalis, but they are insoluble in neutral salt. They are plant proteins uh, that create inflammation and in allergic reactions in our body. Glutalin of wheat, origin of rice. They are the example of glutalin. Next, prolamines are alcohol soluble protein. The prolamines are soluble in 70 to 80% alcohol, but they are insoluble in water, neutral solvent, or absolute alcohol. Example, seen of corn, gliadin of wheat. Next, histone. The histones are soluble in water but are not coagulated by heat. Histones are basic proteins. The histones, being basic, usually occur in tissues in salt combinations with acidic substances such as nucleic acid. Nucleoprotein, example, histamines, histones. Then protamines. The protamines also also have the basic amino acid. Arginine is reached. The protamines are soluble in water but are not heat coagulable. Like histone, they are occur in tissues with nucleic acid. Example, nucleoproteins. Next, we will see about a scleroprotein. Fibrous proteins are also called scleroproteins. They are insoluble, high molecular weight fibers. Example of scleroproteins are collagen, found in cartilage and tendons; elastin, found in tendons and arteries; keratin, of hair, skin and nail. Next major group is the simple proteins. This under all. Next we see about a conjugated protein. Conjugated proteins are composed of simple protein combined with some non-protein substance. The non-protein group is referred to as the prosthetic group. Additional groups. Nucleoproteins, example, the nucleoproteins are composed of simple basic proteins, histones and protamines with nucleic acids. Of nucleic acids means the DNA and RNA. That's the prosthetic groups. They are protein of cell nuclei. As example, nucleohistone, nucleoprotamides. Next group is glycoprotein and proteoglycans, mucoproteins. They consist of simple protein and a carbohydrate as prosthetic group. When carbohydrate content is less than 4% of protein, it is called glycoprotein. That means if the carbohydrate is combined, the 4% of carbohydrate is combined with the protein called glycoprotein. Above this, more than 4% of its combined with protein that is known as mucoprotein or proteoglycans. Examples of glycoprotein: mucin of saliva, immunoglobulins, and mucoproteins. Examples: glycosamine glycans. Next, chromoproteins. Chromo means color. Colored group as a prosthetic group. Example: hemoglobin, cytochromes, catalase, peroxidase. We all know about that. Hemoglobin is a colored protein. Next, phosphoprotein. The phosphoproteins are formed by a combination of protein with a prosthetic group of phosphoric acid. Example: casein of milk, vitelline of egg yolk. Next, lipoprotein. The lipoproteins are formed by a combination of protein with a prosthetic group lipid. Serum lipid lipoproteins. Lipoproteins are useful for carrying the lipid material from intestine to other body and from liver to other organs. Next, chylomicron, very low density lipoprotein, low density lipoprotein, high density lipoprotein. These are all lipoproteins, types of lipoproteins. Next, we see about metalloproteins. The prosthetic group is metallic elements such as ferrous, copper, manganese, zinc, magnesium. These are all prosthetic group in metalloproteins. Example: ceruloplasmin is a copper containing protein. Next, carbonic anhydrase, carboxypeptidase, and DNA polymerase are zinc containing proteins. Next, derived proteins. Derived proteins, also known as derivatives of protein, are hydrolytic products of proteins. These class of proteins are named implies includes those substances formed from simple and conjugated protein, which means when hydrolysis or treatment with any other factors, the proteins, that means the simple and conjugated proteins, degraded or converted into the derived protein or derivatives. There are two types: primary derived proteins and secondary derived proteins. The primary derived protein, also known as denatured protein, which means the proteins, the normal structure is changed, so that is denatured protein. These proteins derivatives are formed by agents such as heat, acid, alkalis, etc. These are synonyms with denatured protein. Example: proteins, metaprotein. The proteins, earliest product of protein hydrolysis by action of dilute acid or enzyme. These are the factors. Example: myosin, myosan from myosin, fibrin from fibrinogen. Next, metaprotein. The metaproteins are formed by further action of acid and alkalis on proteins. Acid and example: acid and alkali albuminates. Then we see about secondary derived protein. Secondary derivatives are formed in progressive hydrolytic cleavage of the peptide bond. In first, the all other bond are removed. First peptide bond is not removed, but here the peptide bond also removed into progressive smaller molecules. Example: proteases, proteoses, peptones, and peptides. This is the flowchart denotes the derivatives of protein or denaturing of protein. Protein convert into proteins, proteins convert into metaproteins. Both are under primary derivatives. Next, the metaprotein convert into proteoses, and proteoses converted into peptones, peptone convert into peptides. These are secondary derivatives. And finally, we get the amino acid. The amino acids are the basic unit of protein.